Hello, I'm Karen Hornby and I'm welcoming Bev Goodyer and Alex Jenkin to this Q&A to answer a few questions that have come out of the Teaching Practical Biology course about inspiring students with plants in science. But first, do you want to say a few words, Bev and Alex, about the course in general?
Well, I suppose first I'd like to say thank you to everyone who's done the course. It's the first time we've done something like this, and it's been really enjoyable for us interacting with people. We've learnt a lot from people on the course and seeing how they've reacted to the things that we've suggested. And it's felt like a very, very positive experience. So I hope everyone's enjoyed it, but it certainly feels like it's been a good experience on our end. And there are some things that we're going to, giving us some food for thought to try out as well, some sort of hints and tips for practicals that we need to investigate a little bit further before we can start putting them into the resources we put on our website. But yeah, it's been great.
That's good that it's been a sort of a two-way process as well, so feeding into future development work.
Yeah, absolutely. Brilliant. Yeah.
Great. Bev?
Yeah, well, I've really enjoyed being part of it. I've never been part of anything like this before. I really enjoyed the way all the teachers and technicians and everybody taking part in the course were inspired by each other's ideas as well as the ideas that were present on the MOOC. I just thought it was brilliant the way everybody got excited by each other's ideas and I feel it's been a very positive experience, and I hope all the participants feel it has as well.
Yeah, there were some really, really good ideas shared, I thought. We'll move to the first question from Howard, which is really, he was asking, where do classroom teachers turn to for credible explanations to difficult questions and concepts?
Come to SAPS. We do offer an Ask the Expert service, generally for teachers and technicians rather than for students, although students do get in touch with us from time to time. The contact details are all available on our website. So there's a phone number and an email address. We try to get back to people within a week. That's possible. So that's for questions about practical protocols, sourcing equipment, but also about plant science queries around subject knowledge, I suppose. We do also have an archive of previous questions on our website. So if you go onto the SAPS website in the area under Associates, you can go through and you can browse previous questions that other people have asked. So you can have a look there as well. I think just to add one other thing I suggested, do obviously always come to us. We've got a panel that we can ask further questions to as well. But if you're on Twitter, there are lots of friendly researchers out there who may well be able to offer some insight into your question as well. Yeah.
Thank you. Ruth has asked whether it might be more successful to compare and contrast plant and animal material and highlight to sort of compare and contrast the processes that are going on. Bev, have you got anything to say about that?
Well, I think in a way that the MOOC course was born out of a need for us to remind people that they can teach so much of many of the concepts that they're teaching in animal biology through using plant material. So what I hope the MOOC has done is to provide examples of plant-based investigations that can be used in the classroom, so that when people are planning schemes of work and thinking about how they're going to teach a topic, they will actually draw on perhaps their first thoughts, which might be animal-based, but we've now provided a course with examples of how they can actually use plants so easily and so cheaply and ethically in the classroom as an alternative, and also as a unifying concept. These are fundamental biological principles. So I hope that in a way our course didn't need to show the animal parallels, because what we're trying to do is to say, yes, but look how you can use plants to illustrate that same point. And of course, I know the first session was osmosis, and we used plants to say, wow, we were trying to say something that you might not think is exciting is, but certainly in the respiration and the stem cells, I think our overriding aim there was to try to make people realise plants are very useful organisms that you can use to get first-hand practical experience of unifying biological concepts. So I hope we've done that. I hope you've been inspired. Ruth, I hope you're able to take what you've learned from the course and sort of interleave it with your animal examples that you brought.
Thank you. A few questions now about the cauliflower cloning experiment. There seem to be quite a number of technical questions. So from Obi, he has trouble getting access to the nutrients for the medium. And a few people have commented as well that they've had issues with contamination, for example, or the explant's just not growing. Alex, have you got any help for them? Or
so yeah, this the origin of this protocol, as I think it explains in the protocol itself, is that this is a technique that Q uses when collecting plant samples in the wild to bring back to propagate on. So it has been developed with that, or developed from that process of scientific rigor. It's as risk-free in terms of contamination as possible, but contamination does happen. And I would reassure people that that's a normal thing to happen. And it does provide opportunities to think about, well, why might that have happened? What could be the conditions? It's tricky in a school environment to control the environment. If you've got vents that are blowing air around the room, if you've got heaters that are blowing out air, then it's best to turn them off if you possibly can. If there's windows open, they should be closed. But you've also got 30 students moving about in a room, and as much as you encourage them to move slowly and carefully and not lean over their little pots of media, but it still does happen. We sort of would say that actually, if you're getting a quarter of them contaminated, that would still be it's not something to worry about in terms of the protocol. That would be if all of them are getting contaminated, you might want to go back and look at the technique, both for preparing the little pots of media and also for what the students are doing. So, because it's possible they could get contaminated before the students are even looking at them. So it's worth going back and looking at that. And you can, again, like I said, you can use that as an opportunity to discuss what's going on. What is the might contamination be coming from? In terms of the medium, again, this is the recipe that we give is the one that Q uses. People have used other things. I've been in conversation with someone who has used I think it's SAPS solution, which recipe for which is on our website, but it's a bit can be complicated to prepare. And adding that to the agar and Milton tablet mixture to create a medium, but without the kinetin, without the hormone present, and without all of the nutrients necessary that are in the mix that we suggest, you may find that you aren't able to get a longer term growth of the explants. It's something that we think we might be able to have a look into. We might talk to Q about whether there are any alternatives. But in general, I I mean, obviously, if it's possible to use what we've recommended, but you can try other things. It's just you might not be able to get any long-term growth. You might find it's a bit like keeping a cut flower in that it stays alive but doesn't develop any new green leafy parts of the plant, which is the real wow bit of that practical, is when they start growing little leaves and looking like little plants rather than little chunks of cauliflower. So I hope that's useful. Again, people are more than welcome to get in touch with us via the contact page on our website, and we can work through any specific issues that people might be having.
We've got some queries about the garlic bulbs as well for the root tip squash. Is it better to use old garlic, new garlic, put it in the fridge first? All kinds of ideas have been thrown into the mix, but what do you recommend?
I mean, my experience is anecdotal. We've been doing we do this sort of every so often. I wasn't involved in the development of this practical. Generally, most garlic bulbs should grow. I do know a technician who absolutely swears by using older bulbs that have been hanging around for a while, just in the cupboard. She finds that works better. The suggestion of putting them in the fridge relates to winterization or vernalization of bulbs. So some bulbs will only germinate or only start growing, after after a period of cold. But I don't know if that's true of garlic. So again, I think it's another one for us to investigate, or if anyone's done any of their own investigations and thinks that there's something that makes them grow particularly, then do get in touch. Another thing we do is sometimes actually putting them in slightly warm water to grow can encourage them to grow a bit faster. So putting them in a water bath at 20 degrees or something can help them along.
Okay, thank you. Bev, anything to add?
Well, not so much for rooting the garlic. It's not something that we've ever had a problem with. They always root well. The thing that we used to have a problem with was the first practical of the day on a cold winter's morning. It would be quite common to find no dividing cells in the root tip squashes we prepared, whereas later in the day there'd be plenty. Now, I have heard that there is a diurnal rhythm for the cell division, but we also wondered whether it was just perhaps the fact that the roots had been sitting in very cold water because there'd been no heating on in the prep room overnight, whereas later in the day they were warmer. So we always used to just make sure that the water that the rooted cloves were sitting in was sort of room temperature or thereabouts for about half an hour before the practical actually started. And that, in my experience, produced dividing cells, mitotic cells on each of the slides that the students made. So just a thought, but anecdotal.
Yeah, a nice bit of advice though, Bev. And the last one, several people have asked this: why do we use the glass beads when we're germinating seeds in the flask?
Right, shall I start with this one, Alex?
Yeah, absolutely.
Well, you can do the peas in the thermos flask experiment with two flasks. You can do it with germinating peas and with cooled, boiled, germinating dead peas. You can do it that way. The flask with the glass beads in is there as a control, because if your hypothesis is that the heat being produced is the result of respiration and you get heat in your glass bead flask, then clearly glass beads don't respire, so therefore your hypothesis is not being supported. So it's there as a control. In terms of using glass beads to keep the volume constant, that was obviously something that we did for the respirometers, because you're dealing in changes in gas volume. And inside the thermos flasks, obviously changes in gas volume aren't as critical because you're looking at changes in temperature. But it's just another way of trying to control the variables. If you've got the same volume of peas in each flask, then you've got the same volume of peas potentially respiring and producing heat to fill the volume of the flask evenly or raise the temperature of the air inside the flask. So it's, from my point of view, it's there as a control.
Well, thank you to both of you. It's been a real pleasure reading all of the discussions during the MOOC, also following up on some of the great resources that people have shared on there as well. So I think that was a particular favourite part of mine. So thank you to both of you.
Oh, thank you very much. Thanks for your support as well, Karen, and Matt as well. Can I just add something on the end, a sort of a what next for those of you who finished the course? We do send out a newsletter every half term, so please sign up for the SAPS newsletter on the SAPS website, and do tell people about the course and tell people about SAPS as well. We're here to support teachers and technicians, and the more we can do that, then that's fantastic. And again, thanks for doing the course, I suppose.
Thanks, Alex.
And the same from me, and thank you to everybody who shared their enthusiasm. Keep inspiring, that's what I would say. So thank you to all the participants, and thank you to you and Matt for all your patient and helpful support throughout.